As we recall from Chapter 12, Section 1, DNA structure, DNA is made up of deoxyribose, sugar, and phosphate on the sides, and in the middle are your nitrogen bases, which are adenine and thymine, which always pair together, and guanine and cytosine, which always pair together. Now it's a two-stranded molecule, and it's held together by hydrogen bonds. So today, we're going to kind of talk about a brief description of the chromosomes in your DNA, and then how replication occurs. So when we're talking about chromosomes in your DNA, we talk all the way back to Chapter 7 when we talked about the difference between a prokaryote and a eukaryote. Prokaryotes are the small organisms such as bacteria. We know that they don't have a nucleus, so their DNA is just located in the cytoplasm. They have a single DNA molecule containing nearly all of the genetic information that it needs. So here's an example of the E. coli bacteria. Now it's still the same structure. You see it's a two-stranded molecule up at the top. You see the cytosine is pairing with the guanine, adenine is pairing with the thymine all the way down. So they still have the same basic structure of the DNA. They just have less than we do, and it's not located in the nucleus. So, like ourselves, humans and other animals, we are eukaryotes. We have about 1,000 times the amount of DNA as prokaryotes, so we have much more. Now, when you get into more about this, all of our DNA does not code. We have some parts of our DNA that doesn't code for anything. And as we know, your DNA is located inside the cell's nucleus, and it's coiled, and it makes chromosomes. We've talked about it before that chromosomes vary between one species to the next. Some can have a lot of DNA, and others have very smaller amounts, but it does vary. Here's a structure of your DNA, like we just talked about. So your DNA, it coils tightly around histones, and then that's what is going to form a chromosome. Last chapter we talked about chromosomes are only visible in cell division. Usually it's the uncoiled DNA. Which now brings us to DNA replication. Each strand of the DNA double helix has all the information needed to reconstruct the other half. So what ends up happening is that half of the DNA strand, it's going to split, and it just forms two brand new strands. Here's a brief picture down at the bottom of how that is going to take place. DNA replication occurs in hundreds of places. Your DNA is so long that if it only took place in one place, DNA replication would take way too long. So this DNA replication can take place in many places on one DNA strand. It's going to go in both directions until that entire chromosome or DNA strand is copied. Now, if you've ever heard of like a fork in the road, okay, the reason that's called that fork in the road is because it's going to start at one and then split into two directions. Now, this is where we get replication fork. In your DNA, the area that it's splitting, and you can see from the picture right down there in the middle, that's going to be called a replication fork. That's the site where it actually separates, and replication is going to begin. Now, you always have to be able to make new DNA. When we talked about cell division, all of your cells have to divide. If you have a break in your skin, a broken bone, your cells have to divide to fix that. So, to do that, you have to get new DNA. So before it divides, if we recall an S phase of interphase, that's when the cell is going to duplicate, the DNA is going to duplicate, and we call this replication. And this is going to make sure that all cells have the same amount of DNA, and it also makes sure that the DNA is the exact copy of itself. So a key concept is that during DNA replication, what takes place is the DNA molecule separates into two separate strands, and then it makes two complementary strands following the rules of base pairing. Now, when we talk about the rules of base pairing, this is when I say that A has to pair with T, and G has to pair with C. Each separate strand is going to be a template, so it's going to act as a template for the brand new strand. And then after replication occurs, you have 2 exact copies of that DNA. Here's another pretty good picture of it. You see the replication forks on both sides where it's splitting. And then the new bases come through, and that's how you get a brand new DNA strand. All right, a couple enzymes you need to know about and how this occurs is we talked about the 2 strands of DNA are held together by those hydrogen bonds. Now we have to we call it unzipping them, and the enzyme that does this is called the DNA helicase, and that's going to go through and break those hydrogen bonds that are holding the bases together. And this is also going to cause the DNA strand to unwind. Now from there, another enzyme is called DNA polymerase. DNA polymerase is going to come back through and put on the brand new nitrogen bases, or the nucleotides, and this is how you get 2 new strands. And also, DNA polymerase also serves, it's going to proofread to make sure that no mutations are taking place and make sure that your DNA is copying itself like it should. A lot of times if it doesn't, that's going to be when a cell may exit that and not go on because of a possible mutation. So kind of the main points here is that your DNA has to replicate to form new cells. And DNA replication is going to follow those base pairings. A pairs with T, T pairs with G, G pairs with C, to form those 2 identical DNA strands.